Okay, let's start. I'm really sorry for the background noise, but let's just do this. Let's solve this question paper together, and I would suggest that you print this paper out first and then watch this video along with me and solve this paper. Okay, so let's start. Question number 1. Dehydrogenase is an enzyme found in cells such as yeast. So we're talking about yeast. Methylene blue is an indicator that can be used to show the activity of dehydrogenase. In the presence of active dehydrogenase, methylene blue changes color from blue to colorless. A student carried out an experiment to investigate the effect of temperature on the activity of dehydrogenase using yeast suspension, that is basically yeast mixed with water. Now they carefully measured 5 centimeter cube of yeast suspension and placed this in a test tube labeled A. The student also measured 5 centimeter cube of yeast suspension which had been boiled and then cooled and placed this in a test tube labeled B. Now both test tubes were placed in a beaker of water kept at 25 degrees Celsius. After 5 minutes, the student added 1 centimeter cube of methylene blue solution to each test tube and mixed the contents. Then every minute for 10 minutes, so that is important, every minute for 10 minutes, they observed the contents of the 2 test tubes and recorded their observations. Now, the student then repeated this experiment at a temperature of 35 degrees Celsius. Okay, so now as you can see in the table, we have 25 degrees Celsius and we have 35 degrees Celsius. For each of the temperature, we have both the test tubes. Now the tick mark indicates the presence of a blue color. That is, the methylene blue did not change any color. And the absence of a blue color means the methylene blue changed its color. So it became colorless. Okay, so this actually means that we have active dehydrogenase. Okay, and this means it's inactive when it's blue color. Okay, now as you can see a difference in each case. In test tube A, we actually have the absence of the blue color. Now in the case of 25 degrees Celsius, the absence started quite late. একটু দেরি করে স্টার্ট হয়েছে, দেখতে পারতেছো যে 8 মিনিট থেকে আমরা কালারলেস পাওয়া শুরু করছি। and 35 ডিগ্রি সেলসিয়াসের সময় আমরা 5 মিনিট থেকে কালারলেস পাওয়া শুরু করছি। দ্যাট মিন্স 35 ডিগ্রি সেলসিয়াসে আরো আগে আমরা অ্যাক্টিভ ডিহাইড্রোজেনেসের টেস্টটা পাইছি। ওকে, that's it. Now let's move on to the next page. Okay, now explain how you would be able to check that the temperature of the water in the beaker was at 25 degrees Celsius. How would you check a temperature? Obviously with a thermometer. So simply, because it's 1 mark, you can just simply write use thermometer. Next, describe how you would adjust the temperature if it became higher or lower than 25 degrees Celsius. So, if the temperature is greater than 25 degrees Celsius, we would add ice, for example, to basically cool it down. And then if the temperature is lower than 25 degrees Celsius, we would apply heat because we want it to be at 25 degrees Celsius. Okay, that's it. Next, Explain why the test tubes were left in the beaker of water for 5 minutes before adding methylene blue solution. Why did we wait? Why did we wait when we placed the test tubes in the beaker of water, which was at 25 degrees Celsius, and then we waited 5 minutes before adding the methylene blue solution? Because we wanted the test tubes to actually reach 25 degrees Celsius. আমরা তো একটা বিকারের মধ্যে, যেটার মধ্যে ওয়াটার আছে 25 ডিগ্রি সেলসিয়াস ওয়াটার, সো ওটার মধ্যে আমরা দুইটা টেস্ট টিউব সাবমার্জ করতেছি, ফর এক্সাম্পল. Okay, this is our test tube, this is our test tube. Sorry for the bad drawing, but I hope you understand. And then we would wait for the test tube to get to 25 degrees Celsius, right? So, to allow the test tubes to reach 25 degrees Celsius. Okay, that's it. Next, using the information in table 1.1, describe the student's observation. The previous table they meant. So you can go back just the previous page and check. I already showed you that 35 degrees Celsius আমাদের test tube A তে 5 minutes থেকে colorless হয়ে গেছিল, methylene blue. And 25 degrees Celsius 8 minutes থেকে আমাদের test tube A colorless হয়ে গেছিল. Okay, so how would you write it down? So in test tube A at 25 degrees Celsius, the solution became colorless at 8 minutes, whereas at 35 degrees Celsius it became colorless at 5 minutes. Okay, and in test tube B we did not actually see any sort of color change. So in test tube B there was no color change. Okay, now you have 3 marks over here. I'm going to point it out with red points, red marker. So for this one you have 1 mark, for this one you have 2 marks, and for this one you have 3 marks. Okay, now suggest an explanation for their observations. Okay, so now very important, this question asked just to describe the observations, not to interpret it. Do not interpret here. Do not give your own opinion je, oh maybe এর জন্য হইছে, or maybe এটা হইছে দেখে colorless হইছে. You don't even need to do that. You just simply describe. That is, you say what you see. What did you see on the table? Just write it down. Okay, do not interpret. Here you have to interpret. Okay, so interpret the table. Suggest an explanation for their observations. Obviously, because test tube B, which had no color change, there was no active dehydrogenase. So in test tube B, there was no active dehydrogenase. Okay, that means that basically indicates, not means. That indicates the yeast were, you can say dead, or I would say inactive, because that sounds better. Now in test tube A though, the enzymes were actually working. That's why there was a color change. Okay, so in test tube A, the yeasts are active. Okay, now you can also write it down that in 35 degrees Celsius the activity started quite early. Okay, there was enhanced enzyme activity. Why? How do you know this? Because আমাদের color change টা আগে হইছে. The color change occurred at 5 minutes. Okay, so there was an enhanced enzyme activity. That's it. That's where your 3 points go. So I would point it out. First thing is this one. This gives you 1 point. This one gives you 2 points. This one gives you 3 points. Okay. I forgot a full stop over here. Okay. Now, suggest one way in which this experiment could be improved to increase the reliability of the results. Now, think of one error or misinterpretation that might happen in this experiment. A very easy one to actually think of is that, I think I'm just going to mention it over here. In labs, we tend to repeat experiments, okay? Why? Because this increases the reliability of our data, the reliability of our results. So this is something you can always write. This is a very easy thing to say, because as you can see in the experiment, they only did the experiment once with 25 degrees Celsius and once with 35 degrees Celsius. They did not repeat the experiment. So you can say to be reliable, the experiments can be repeated for each temperature. Okay, that gives you 1 point. Moving on to the next page. Okay, now okay, giving full experimental details, describe an experiment. Giving full experimental details, describe an experiment you could carry out to investigate the effect of pH on the activity of dehydrogenase. Okay, using yeast suspensions and methylene blue solutions. So in the previous question, you actually saw that the student was trying to experiment or investigate the effect of temperature. And now we have to do basically the same experiment, but with pH. We have to see the effect of pH on the activity of dehydrogenase. So you can simply copy the previous experiment that the student did, and basically instead of working with two different temperatures like 25 degrees Celsius and 35 degrees Celsius, you can choose 2 pH. In my opinion, I would choose in fact 3 different pH instead of 2 different pH, because that will give us a better picture. For this experiment, I can choose maybe a pH less than 6 or 7, because 6 or 7 is neutral, and that is equal to 6 or 7, and maybe another pH that is greater than 6 or 7. Okay, so we have to now play with 3 different beakers, 3 different solutions. Okay, now let's prepare. First step is preparing 3 different solutions with 3 different pH. Okay, so now we will be prepare the yeast suspension, which is a must. Without this we cannot even experiment. So prepare the yeast suspension, add buffer to it as well, because we want that solution to actually, you know what, let's write it down later. We want that solution to actually be in the same pH, so that's why we need to add buffer, because yeast tends to actually produce ethanol and stuff, so our solution can start to get a bit alkaline or acidic, I don't know, depending on what the yeast is doing. So we need to add buffer because we want it to be at the same pH. Now, next point is, just like the previous experiment, add 5 centimeter cube. I think that was the previous question, right? Let's check. They used 5 centimeter cube of yeast suspension, so I'm gonna use the same. Add 5 centimeter cube of yeast suspension. And then we have to add the pH solutions. So So we are adding everything to a test tube. So we are adding 5 centimeter cube of V suspension, a particular pH solution, and methylene blue solution to a test tube. So I think we have to mention that, so we will have 3 different test tubes. Containing the 3 pH solutions. Oh man, I forgot the buffer again. And buffer. Okay, now I'm going to draw my 3 test tubes because I always tend to draw my experiments. So we have this, this, and this. Let's give it different color. Okay. Peach. Let's write 7. Okay. Now, we are done almost though. We need to keep all the test tubes in the same water bath of, let's say, 25 degrees Celsius. So that is a water bath of 25 degrees Celsius. Keep all the test tubes at the same temperature. So the last thing is record the observations every 1 minute for 10 minutes. Okay, that's it. So it consists of 5 points, so make sure you write down everything, and that's it. Now, yeast is used for the production of alcohol in the brewing industry. In an investigation, the production of alcohol by yeast was recorded every 5 hours for 25 hours. The results are shown in table 1.2. So we have the time from 0 to 25, and we recorded the alcohol concentration in gram per dm cube. Okay? All right, let's look at the question on the next page. So here we have to construct a line graph of the data in table 1.2 on the grid below and draw a smooth curve through the points.